Now I'd like to introduce our first speaker, Carrley Smith, who will be speaking about why does the Salish Sea glow at night? Carrley Smith is a recent graduate from Western Washington University in environmental science. As a sea kayak guide, she has led bioluminescence kayaking trips in the San Juan Islands. Oh, how much fun would that be? For the past 3 years. In 2023, Carrley conducted research on bioluminescence in Bellingham as an intern with the Boating Center. As well, Carrley has also studied with algae researcher Robin Kodner to identify the patterns and species of the bioluminescent organisms. Carrley is excited to share recent research on the natural and human histories of bioluminescence locally and globally.
Welcome. Thank you so much for having me. I have a lot of information, some pretty awesome information on bioluminescence tonight. So I'm going to go ahead and jump right into it here. So what is bioluminescence? Bioluminescence is the production of living light. It is a chemical reaction within organisms that produce light. And this is different from phosphorescence, like a glow stick, or fluorescence, like UV light. And I hope everyone has had the chance to see fireflies or lightning bugs, depending on where you're from here. And they're flashing their lights in a specific pattern to try to find a mate. And bioluminescence is referred to as a cold light. And it's cold because it doesn't produce much heat, less than 1%, which is pretty incredible compared to the incandescent light bulbs that we use. And those lose about 90% of energy to heat. So bioluminescence is one of the most efficient forms of light production that we know of on our planet, which is pretty cool. And on land, like the firefly, we see bioluminescence a fair bit. It's pretty common and famous in New Zealand and Australia with the glowworm caves, which hold a glowing larval stage of an insect. And there's bioluminescent mushrooms, which are pretty incredible. I've never seen one, but there's about 100 species worldwide. And there's some that live in the Salish Sea as well. And they were recently thought to glow because they attract insects to disperse their spores for them, which is pretty interesting. And we'll talk a lot about human use and human history with bioluminescence. And for mushrooms, there's reports of people in Scandinavia that would use the bioluminescent mushrooms at night to light their homes. And in Indonesia, people would take these mushrooms and use them as flashlights to guide their way through the dark jungles, which is pretty incredible. There's a lot of bacteria that bioluminesces as well. And there's all sorts of stories of people making a fish dinner, discarding the, fish skins and head in a trash can, and then being pretty surprised when they see that it's glowing after some time. And these fish skins that are latent with bioluminescent bacteria, miners used to bring them down into mines, or they'd bring fireflies to light, their way through mines before the 19th century, when open flame wasn't safe and there weren't safety lamps yet. So that's bioluminescence on land. And nothing compares to bioluminescence in the sea, in the ocean. And so covering 70% of our planet, the ocean is the largest ecosystem, and it holds the vast amount of life living on the planet. And there's a lot going on. When you live in the ocean offshore, there are so many things that are trying to eat you, and there's nowhere to hide. So a tactic to solve this problem that so many animals have figured out: if you go deep enough, you can find a place in the water where there's no light. You can hide. No one can see you. And at night, the line that separates the photic from the aphotic zone, called the edge of darkness, that rises up at night. And all of these animals rise up with it. And they get to the surface water where all the good food is, and then they dive back down, in the daytime. And this phenomena is the largest daily migration on our planet, which is pretty incredible. So with so many of these animals living in complete darkness, something incredible happens. They start to produce their own light. And there's a few studies that have been done, and it's speculated that over 90% of everything in the ocean can produce light, is bioluminescent. And that is phenomenal because if the majority of life lives in the sea, majority of the life that lives in the sea can bioluminesce, then we as humans are in the minority of creatures on this planet who cannot produce light, which is a bummer, I think. And um, as far as bioluminescence goes, it has evolved over 50 times independently. So there's no one bioluminescent family tree, that everything evolved from. It's evolved separately over 50 times, which is incredible. Another thing that has evolved over 50 times on its own are eyeballs, and pretty useful, at least for us, but pretty useful for things in this world. And eyeballs, no matter if you're a fish or a human, they look and do pretty much the same thing. And that's why bioluminescence is extraordinary, because all of these creatures have evolved different chemicals and different ways to produce bioluminescence, and they have all these different ways of using it, and they're pretty extraordinary. And there's all these, different reasons to bioluminesce: to attract prey, to camouflage yourself, as defense against your predators, to mate, to hunt. Overall, it's a form of communication. And because there's so much that lives in the sea that bioluminesces, it might be the most common form of communication on our planet, which is pretty cool. On the right-hand side, this image just shows different taxa that have developed the ability to bioluminesce. And I pointed out a couple important ones. They are dinoflagellates, and don't worry, I will explain what that means. And then at the bottom there are ray-finned fishes and sharks and rays, which is pretty cool. We'll get into some of the cool creatures that bioluminesce and why they do it.
It's so pretty.
I'm feeling happy, which is a big deal for me.
I want to touch it. Oh,
Hey, come back. Come on back here.
I'm gonna get you.
I want to get you.
I'm gonna get you.
Swim with you.
I'm gonna get you.
I'm gonna be your best friend. Good feelings gone.
So good. All right. So that's the anglerfish. And the anglerfish has this famous lure on its head to attract prey. And that lure is called an esca, and it's a little house for bioluminescent bacteria. And some of the bacteria is unique to each species of anglerfish, which is interesting. Anyways, when you are living thousands of meters in the deep darkness of the ocean, you don't come across other fish very often. So when you do, you need this huge mouth to be able to fit anything, inside of your stomach. And you have this lure to bring in prey. And they've also developed a really interesting form of mating called sexual parasitism. And for a lot of these anglerfish, there is a tiny male that finds a big female anglerfish, and he attaches himself to the side of her body. And eventually, they fuse together, connecting their circulatory systems. And the female has full control of when the sperm is released, and she waits until conditions are optimal to fertilize her eggs. But that's kind of an aside. Another form of bioluminescence, a use of it, is camouflage. And this is a really cool one. So a lot of sharks and fish, as I'm sure you are all aware, will have a darker top side and a lighter bottom side. So they're pretty camouflaged depending on if you're looking down at them or if you're looking up at them. And animals use the same idea, but with bioluminescence. And this is called counter illumination. And there's these bioluminescent organs called photophores, and they'll cover the bottom section of these animals. and they have the ability to match with the same exact light intensity the moonlit waters above them, and they can control and change this. And it's amazing. And this shark here is the cookie cutter shark, and the cookie cutter shark does this counter illumination, except for one spot on its belly, which remains dark. And it's thought that when a predator is looking up at it, they think it's a small fish or just prey item. So they'll swim up to it. And then sure enough, there's a cookie cutter shark right there, that has its dinner, and it has the shape of a perfect circle, thus getting its name. So another one is defense against predators. This is the pocket shark, a shark so small it'll fit in your pocket. As the cutest shark in the sea, it looks a little different than other sharks. It was recently identified in 2010, and it's really deep living in the Gulf of Mexico. And it has these bioluminescent pockets full of liquid. Then when it feels threatened, it's hypothesized that it squeezes those out, stuns its predator and swims away. And it was found by folks that were studying sperm whales, which I think is hilarious because it looks like a tiny sperm whale. We have mating displays. These are ostracods. They are tiny crustaceans about the size of a sesame seed, and they're quite famous for their mating display known as string of pearls. And what this is a male ostracod vomits mucus strings with balls on it, and they bioluminesce. And a female ostracod finds one of these, it climbs to the top, and then ostracods do what ostracods do at the top there. And this phenomena is really famous in the Caribbean for scuba divers. And supposedly, these mucus balls are pretty extraordinary when you see them at night all around you. I am saving the best for last, in my opinion, a hunting strategy. This creepy deep sea fish is called the stoplight loose jaw. And if I put a real photo up, it would terrify you all. They're very weird looking. But this fish has blue and green photophores, bioluminescent organs, and whatever. The rest of the fish that we've talked about have the same thing. But this fish also has red photophores next to its eyeballs. And this is interesting because water absorbs red light. And at about 15 feet, red light virtually disappears. At 100 meters down, a red fish, you won't be able to see it. So all of these deep sea creatures, they don't have the ability to see red light because there's nothing red. There's no reason to, except for the stoplight loose jaw. It has these red flashlights that it shines that are invisible to every other creature down there. So it's, hunting with its own personal flashlight, and it has an open connection to communicate with the other stoplight loose jaw, with nothing else seeing it. So, it's like a deep sea ninja, for a lack of better words. So those are pretty cool. And as many of you know, we don't need to go down into a submarine to see these incredible bioluminescent displays. We get to see them right here on the surface in the Salish Sea. You will, if you touch the water in certain areas with your hand, with a paddle, propeller of a boat, you can see bioluminescence and crashing waves. What was mentioned earlier, if there's a fish or a seal swimming through the water. There's a book called Island Fishing by Boyd C. Pratt. And he talks about the Semiahmoo First Nation fishermen just north of Bellingham, Washington, right on the border. And they would paddle out in canoes at night to hunt sturgeon. And they would see the sturgeon swimming through the bioluminescence. They would spear them, connect a line to the spear, and these sturgeon are huge, powerful fish. So they would let them swim around like crazy until they got tired enough, stop swimming, and then they would reel them back in, and throw the sturgeon in a canoe, which is pretty interesting. But there's a quote that I love from the HMS Challenger, which is a British ship that launched in the late 1800s for the purpose of exploring the world's oceans. This expedition was the first of its kind, and it laid the foundation of Western oceanography as we know it. But one of the English crewmen aboard wrote about bioluminescence. He said, "It was as if the Milky Way, as seen through a telescope, scattered in millions, like glittering dust, had dropped down into the ocean, and we were sailing through it." The gal, Lucy Greeley, who started this internship, she says that she loves it so much because it's very unearthly and it's alien-like. And you can see the stars above you, and then you can, see the bioluminescence that look like stars below you, and you can touch them and ignite them. And it instills such a wonder to be able to do that. And even if you go out and see them and you don't have this like deep feeling in your chest, it's so cool to see grown adults just squeal and exclaim like children when they see it. It's pretty fun. But as long as we've been humans on the water, we've been interacting with and questioning bioluminescence. We find writing about bioluminescence over 2,000 years ago in Chinese text, talking about the fiery sparks you see when the water is stirred. Sailors have known it as sea sparkle. Aristotle wrote about it and talked about the little glowing animals in the water and coined the term cold light. Pliny the Elder, a Roman naturalist and philosopher, wrote about finding a glowing jellyfish on the beach and rubbing a stick in the jellyfish goo and using that to light his way along the beach at night. Potentially, there was no light pollution in the first century, so, a little different there, but those are pretty beautiful. Some more recent human history is military and wartime use with bioluminescence. During the U.S. Civil War, there was a major battle, the Battle of Shiloh in 1862. This was a particularly brutal battle for the Civil War, and that says a lot. And after the battle, these troops were left for days in the cold, wet mud of trenches, and they had open wounds. And these open wounds were saturated with bioluminescent bacteria, and their wounds would start glowing. And weirdly enough, the soldiers that had these glowing wounds were more likely to survive than the soldiers that did not. And they dubbed this the angel's glow. And we know now that this bioluminescent bacteria is toxic and it kills more harmful germs. So it was more or less sterilizing the soldiers' wounds. In World War I, bioluminescence became very famous when a British torpedo sank a German U-boat, and it spotted it by its bioluminescent wake going through the water. And this was prior to the advent of LiDAR or radar. So this was a pretty big deal to be able to spot, a submarine or a ship or a torpedo or whatever it was. So most of the research that has gone into bioluminescence has been from militaries worldwide. And if you have a large tanker passing through bioluminescent waters, the wake is so large you can see it from space on satellite imagery. And some World War II history, let me go back. Some World War II history, the Japanese army, they harvested vast amounts of ostracods. Ostracods are those little sesame seed crustacean creatures that have the mating display that we discussed. And these are really interesting because once they're dried, their bioluminescent properties are preserved. And if you add a little bit of water, they'll start glowing. And Japanese soldiers would keep a little vial of them on their person, and they would add some spit, and they would have enough light from these ostracods to be able to read maps, letters, or they would paste them on the back of their helmets. And at night during combat, they could see each other and see who was friendly, but it wasn't enough light to give away their position. A Japanese scientist by the name of Osamu Shimamura changed biology and medicine forever when he isolated the green fluorescent protein, or GFP, from jellyfish off of San Juan Island. And Shimamura lived outside of Nagasaki at the time that the atom bomb was dropped, and his grandma promptly threw him in a bathtub. And he believes that that may have saved his life from radiation poisoning, but he eventually came to study bioluminescent proteins. Him and his family moved to Friday Harbor for the Friday Harbor Marine Labs on San Juan Island to study the crystal jellyfish, or Acora victoria, or water jellyfish, whatever you call them. You'll see them in the summertime. You know, they're everywhere. You can pick them up too, which is pretty cool. But they came out to collect these jellyfish. Eventually, they collected nearly 1 million of these jellyfish, supposedly hiring local kids about a cent per jellyfish, but that's not fact checked, so keep that in mind. And these barrels of jellies were compressed like apples or olives, and you would end up with this, luminescent goo. And with this goo, he was able to isolate one of the proteins that glows under blue or ultraviolet light, and he called it the green fluorescent protein, or GFP. And scientists use this protein to tag the once invisible proteins or genes. And if you tag these genes with GFP, if you hit them with a UV or a blue light, then they glow. So you can visually see if a protein or a gene is being expressed. And this discovery was so transformative and indispensable to biology and medicine. One gal I was talking to said it was akin to the invention of the microscope. This was a pretty big deal. And in 2008, Shimamura and his team won the Nobel Prize in Chemistry for their work on GFP. So I have one last story for you all, and that is about James Lovell. He was an astronaut commander on the Apollo 13 mission. It was NASA's third trip to the moon, or almost to the moon, at least. There's a movie about it, go watch it. But before Lovell was an astronaut in this, historic event and whatnot, he was a fighter pilot, and he owes his life to bioluminescence. In 1954, 1 year before Osamu Shimamura began studying bioluminescence, Jim Lovell was on a training mission off the coast of Japan. He was flying his airplane at night, trying to land on an aircraft carrier, and he got caught in a storm. And for whatever reason, all of the instruments in his plane and in his cockpit went out. It would just, everything stopped working. So he was flying blind at night in a storm, trying to land on an aircraft carrier that he could not see and could not find. So he was screwed. But that was until he noticed a faint green shimmer of the bioluminescent wake that the airplane or the aircraft carrier was giving off. And because of the bioluminescence, he was able to land safely and then go to the moon, or almost go to the moon. So the question remains, what saved Jim Lovell? What is this glowing water all about? What causes this phenomenon? And this phenomenon is caused by plankton. What are plankton? Plankton are drifters and floaters. They are creatures so small that they're unable to swim against the current. There are two types of plankton. There are phytoplankton, which are the little plants. And, they eat sunlight and turn it into sugar and do their photosynthesis thing. And then there are zooplankton, which are the little animals, and they eat the photosynthesizers. They eat the phytoplankton. And, little fish eat the zooplankton, and then it just goes up and up and up the food chain from there. And we're going to focus on phytoplankton here for a second. And good things come small. I want everyone to take a deep breath with me. About half of the oxygen we have in our atmosphere is produced by phytoplankton in the ocean. There's a phrase I love, that every other breath comes from the sea. And phytoplankton and plankton as a whole play a really important role in our Earth's carbon cycle as well. When plankton die, they sink down into the depths, and this phenomena is dubbed marine snow. But their carbon is either recycled, back into the biologic world, or it's sequestered from the atmosphere. And this process is called a biological pump, and it is responsible for the Earth's largest carbon sink. And about 40% of the carbon emissions since the Industrial Revolution have been sequestered in the ocean by these plankton. So thank you, plankton. As far as phytoplankton go, there are two types of them. And all of these guys are single-celled organisms. And one of the types of phytoplankton are diatoms. Diatoms are the main photosynthesizers. They're the main oxygen producers. And they're made out of the same thing that glass is, silica. And they come in all sorts of these cool shapes and different sizes. But with ocean acidification, silica is dissolved more slowly in the water, so it makes it harder to make new silica shells, so resulting in less diatoms, which, can't be good. But diatoms, when they're fossilized, they're called diatomaceous earth, which you may have heard of. And you can sprinkle diatomaceous earth on your counters if you have an ant problem, then it'll solve that problem. And for larger problems, diatomaceous earth is an ingredient in dynamite as well. Dinoflagellates are the other phytoplankton. I know, what an unfortunate name, dinoflagellate, but dino means whirling, and flagellate means flagellum, or a little tail that they use to propel themselves and move themselves around. And these are all single-celled organisms. The dinoflagellates, some of them eat other things, some of them photosynthesize, some of them do both. They're very weird. But the dinoflagellates are the famous ones because they produce bioluminescence. This was a video from 2020 in California, right when the COVID pandemic caused a lockdown. There was glowing waves, so that must have been interesting. But they also cause red tides or harmful algal blooms. And if you haven't heard of a harmful algal bloom, you probably will here soon. But these guys can be toxic in large quantities, and they can cause shellfish poisoning, big fish die-offs, but they're also so pretty, so. And why does it happen here? Why does the water glow here and in California? And it happens in the Indian Ocean, and it happens famously in Puerto Rico and all over the world in some places and not in others. And as far as the Salish Sea, it is only here in the summer months. And that's because we have a lot of sunshine and a lot of nutrients. As you all know, our water is green in the summertime, and that's because of all of the algae and these phytoplankton blooms and just everything that's living in the water. And as a lot of you probably know, we have some pretty wicked currents in the Salish Sea. And because of this, you get this phenomenon called upwelling, which is where deep, nutrient-rich, cold ocean water rises to the surface. So it brings all of this food up. So it's just a lot of nutrients, a lot of sunshine. And the best time to see bioluminescence is in the warmest summer months and in a protected bay that's not moving around that much. On a windless, still, moonless night would be the best time to see it. And it is only at night you'll be able to see these guys as single-celled organisms. They have the ability to know when it's nighttime and when it's daytime, and they only they only bioluminesce at night. And they have a circadian rhythm, which is what that's called, is when they know the difference. If you got a water bottle full of the bioluminescent water and you shook it up in a dark room during the daytime, they're not going to bioluminesce until you shake it up in a dark room at nighttime, and then they will bioluminesce. So they know, which is pretty interesting. And why do they do it? These single-celled dinoflagellates, why do they light up at night when they're touched? We do not know. Short answer, we don't know. It's magic, I think. But we have some theories, of course. We have theories. So just like all the other creatures we talked about for so many others, it's a form of communication. So most likely it's a form of communication for these guys as well. And these are microscopic creatures. I have to use a microscope to see them. But at night, when you move your hand through them, they light up. They're right there. They're, flashing, and you can see them pretty well. So they light up about 100 times their size. And it's possible that this is a defense mechanism for them. If you tried to eat me and I lit up 100 times, I say 1,000, 100 times, and I light up 100 times my size, I might scare you off pretty good. But I'm also giving away my exact location, which may not be so great. So there's a theory called the burglar alarm theory. And what this is if there's a predator that's trying to eat some of the bioluminescent dinoflagellates, the predator swims through the water and they all light up the body of that thing, potentially in the hope that something bigger comes and eats it before it eats them. So that's a burglar alarm theory. Throw in, your own theories into the chat, because we don't know. But these are also toxic cells. They, emit toxins. So it's potential that they're flashing because they're saying, I'm toxic. You don't want to eat me. So as far as the specific species of dinoflagellate that is causing this phenomena, that's causing the bioluminescence in the water, this one right here is credited globally for producing bioluminescence. And this is called Noctiluca, Noctiluca scintillans, and it's known as sea sparkle commonly. But noct means night, luca means light, and scintillans is to scintillate. So its name literally translates to night lights, that sparkle. There was a study that came out in 2019 that said that the Noctiluca in the Salish Sea and other populations around the world have lost their ability to bioluminesce. So it's potential that they are not bioluminescing in the Salish Sea, and they're still bioluminescing, and the coast of Japan or other parts of the world. So if that's the case, if it's not Noctiluca, then who is it? But I forgot about this first. All right. So the reason that Noctiluca isn't bioluminescing is for this might be a little far-fetched, but pretty much it has the lactose intolerant form of bioluminescence. And what I mean by that, if you're lactose intolerant, you're not producing enough of the enzyme lactase to break down lactose. It's the same thing for bioluminescence, but with these different chemicals. There's a protein called luciferin and an enzyme called luciferase. And you need those two things to produce light, to produce the bioluminescence. And it's thought that the Noctiluca in our waters is lacking a working form of that protein and that enzyme to be able to make this reaction. And just the root there, Lucifer, if you notice that, named after the fallen angel of light. But if it's not Noctiluca, then who is it? So that kind of leads me into the research that has been done at the Community Boating Center in Bellingham Bay in Washington State. And Lucy Greeley, who started the internship, kind of came up with this idea. And on the bioluminescent sea kayak tours, we would paddle out at night and guide these tours, and we would bring with us a plankton net, and that's our little route here. It's about 2 miles from Bellingham Bay down to this protected bay that we dubbed Bio Bay. I know, very original there. But we would bring this net called a plankton tow, and we'd drag it behind our kayaks for about 5 minutes to collect the water, that contains phytoplankton, and then bring it back to the lab and look at it under a microscope. It works only if you attach the tow to yourself. There was a night when I forgot to do that, and the net laid on the bottom of the bay for about 5 days, but that's an aside. So as far as what we found, when we were out there at night, we were taking environmental data as well. So on the wind, air temperature, on sea conditions, anything that we think may have an effect on the bioluminescence. And Lucy in 2022, she took environmental DNA samples as well. So you sequence, the DNA that's present in the water. And she found Noctiluca present in every one of her samples. She also found 4 other known bioluminescent dinoflagellates in her samples. And she found that the air temperature affects the amount of species that are present. And for this last summer, for my research, I took a observation scale between 1 and 10 every night on how well I thought the bioluminescence was. And a friend of mine put it Carrley's subjective scale of awesomeness. And my subjective scale of awesomeness matched up pretty well with things that we already know about bioluminescence, that it's better when air temperature is, higher, and you can see it better when there's less wind. So my scale of awesomeness, has some ground to stand on here. I'd like to mention that none of my graphs are statistically significant, so please don't pay too much attention to them. And as far as things that were found, I found pretty much the same thing. I found Noctiluca cells and 4 other bioluminescent dinoflagellates in the water as well. And, we don't know which one is responsible for bioluminescence. If it is Noctiluca, if it's one of the different species, we don't know. And it kind of gets into the nitty-gritty here, but this population of Noctiluca that's known to be not bioluminescent, it's wrapped up in this California current here that hits the Salish Sea. So it's potential that there's some populations that are bioluminescent, some that aren't, that are in our waters. We don't know. This is a photo of the cell Noctiluca, and I found it in about half of my samples. And when there's big blooms of them, they are toxic. Another one that I found, the serradium, you can see the size difference. You can see these cells here compared to the size of the Noctiluca. They're quite large compared to other, microscopic organisms. But these guys I found in a bunch of samples. These are Alexandrium. They cause paralytic shellfish poisoning. So listen to those signs on the beach that say, don't eat the shellfish. One of my friends can attest to that as well. This is a different species that causes red tides that are bioluminescent. Protoparadinium, common one, they're toxic, and I found them in every one of my samples, and they're also bioluminescent. And so as far as all of these bioluminescent dinoflagellates go, you can buy some online. You can order them, and that's, kind of interesting. But as far as all of this goes, is it important which species of dinoflagellate is bioluminescing in our waters? Maybe, we thought it would be interesting to look into, but overall, probably not. As you know, we have changed our oceans more in the last 60 years than we have in the last 250,000 years since we've been a species. We're changing the temperature, the chemistry, pulling out all the fish, we're putting in pollutants and all this other stuff. We're exploiting this place pretty heavily. And this bioluminescence, it reminds us how magical our world is, and it shows us that. There's the groundbreaking oceanographer Edith Witter, and she's a quote from her book The Edge of Darkness that I love. Our survival on this planet depends on fostering a greater sense of connection to the living world, and wonderment is key to forging that link. And this bioluminescence, it shows wonderment. So I think that's the most important part of all of this. And the Community Boating Center in Bellingham, Washington is a place that is instilling wonder, bringing the community, kids, underrepresented groups onto the bay, having fun, interacting with the environment. And that's kind of what this is all about. that's the end of my presentation. I wanted to thank, the Community Boating Center, my advisor, Dr. Robin Kodner, and the Transition Salt Spring for hosting this. Pretty incredible. One more thing here, please hire me. I am very unemployed and just love a job. And I have some sources down here. Reach out to my email if you want to see any of my other sources or anything. And then this is a video of some of the dinoflagellates swimming around in the microscope. If you haven't learned anything from this presentation, please take away that this is one drop of water that's showing here on the screen. All of these little creatures are in your hair when you get out of the ocean, so take a shower, But yeah, that's all I have. So I can open it up to questions if any of you have questions.
Wow, that was wonderful, Carrley. Thank you so much. It opened up a whole lot of, hopefully a whole lot of questions, but also a whole lot of wonder in everybody. Yes, if you want to go ahead and stop sharing your screen.
Yeah,
and we can go to questions.
sounds good.
Yeah, I've been keeping track a little bit of some comments, lots of great positive ones from people who love the presentation. And I agree, that was really great, Carrley. I've interacted with these little dudes myself and it never gets old.
Right.
There aren't any questions here right yet, but there were a couple comments that people made. I'll shoot those out there. Just some cool comments. Like if anyone, instead of a question, maybe if anyone has a cool story that they have with an interaction with bioluminescence, that might be cool to share. One person mentioned they were in Jamaica swimming and it was only really shallow water and something zoomed past really quickly and it was a bit scary, but they lived, so I'm sure it wasn't that scary. It didn't taste anyone. And then someone else also mentioned that they were doing a kayak tour in the, I don't know how to pronounce it, Viquez mangroves in a lagoon there, and they saw lots of great stuff too. Oh, there's one question here that just came in. It says, enzymes do function based on temperature. Do you think there's a temperature range for Noctiluca?
Yeah, there is a temperature range, and it's a pretty impressive one. They can survive in the Arctic and they can survive at the equator. So they have this range of, just 30-degree water to 70-degree water where they can survive. Yeah, so I don't know.
Well, another question here is, have you noticed any decrease in bioluminescence over the years?
Only an increase, and there's a lot of literature from around the world, in China and Japan, and that there's only an increase in bioluminescence due to these big blooms of algae that we're seeing. But in some areas, you'll lose it as well. For example, touch stimulates the bioluminescence, but a little bit of acid also stimulates it. So in a lab setting, you can add a drop of acid and it'll light up one of the dinoflagellates. So in places where you see a lot of ocean acidification, it'll start to decrease the amount of bioluminescence. So yes and no.
Yeah, I'd say some of our nutrification that's happening with our more extreme weather, washing more nutrients off the land will increase. Like there's a nutrification is a pretty big deal across all over the world. So if that's what plankton wants, they'll show us that they're doing well. Another question. Someone said, did the Noctiluca in the Northeast Pacific used to show bioluminescence? And if so, why did they stop? You kind of touched on that earlier.
Yeah, we have no idea. We used to think because they bioluminesce everywhere else that they bioluminesce here because they're present here. And it wasn't until recently where you were able to test populations and test their enzymes when they found out, oh, they don't have the enzyme you need to bioluminesce, so they must not be able to bioluminesce. But long story short, we need more research. We don't know.
Someone else has already also asked, are they only in the ocean, not in freshwater?
Yeah, only in the ocean, not in freshwater.
There is freshwater plankton, but not as sophisticated as the saltwater.
I don't know if it's sophistication, but none that bioluminesce. Yeah.
And someone else has, how much research is happening on them?
I wish I knew the answer to that, but only more, especially with the amount of nutrification and changes in plankton populations. There's only more happening, but not enough, I think, probably with everything too. But
There's lots of good comments in here, and someone is saying that you have a skill that's been lost, that you're an amazing storyteller and scientist. So any employer would be fortunate to have you. So if there's a potential employer listening tonight, currently there are no other new questions in the chat, but please feel free to enter them. We do have time for several more. I have a question for you in the meantime. You mentioned toxicity and bioluminescence kind of hand in hand. Are they always like does everything that bioluminesces, like that are the phytoplankton, are they all toxic?
That's a good question that I don't know the answer to. A lot of them are toxic, but in such a small amount. They're not toxic to us. If we go out and swim in the bioluminescence, we're not going to be hurt in any way, but they may be toxic to the other microscopic organisms that are trying to eat them. And as far as the ones that are very toxic and harmful and cause the red tides and paralytic and diuretic shellfish poisoning, things like that, some of them are bioluminescent and some of them aren't. I don't think there's a correlation, but I don't know. But good question.
Okay. Well, thank you for that. Yeah, it's just an amazing phenomenon. And do remember, people, during our break to put your bioluminescent stories into the chat. We've had quite a few already. One person saying that we got a course on a guided kayak trip in Costa Rica, trying to find our way back in the dark and relying on bioluminescence to alert us to saltwater crocs.
Yikes.
Yikes. Right.
Yeah, so I think almost everybody has a little bit of a story or some kind of an experience with bioluminescence at some time.
Someone is asking, could you tell us a bit about your night kayak trips, Carrley?
Yeah, absolutely. There are a lot of companies along the San Juan Islands and throughout the Puget Sound and Salish Sea that have tours. And then I've worked this last year with the Community Boating Center, again in Bellingham, Washington, and they have tours, and other companies do too. And if you haven't seen it, it's a great way to see them by boat. And yeah, pretty special.
I've got a bioluminescent story. If no one else wants to share one. When I was an undergrad, I did, friends of mine had to go do a nighttime beach seine. So I'm not sure if everyone here on the call would be familiar with the beach seine, but it's this giant long net that touches the bottom in the intertidal area, and you do it by hand. So you wear your hip waders and you walk out so far and you make a scoop, and then you pull it into the beach and you see what's there, and then you quickly quantify and then release most things. And we did it at night to do a comparison between day and night in this particular area. And I was standing waist deep in water and I could see my feet. My whole body was lit up while we were doing the work. So it was my very first exposure to it. Wow. And been pumped about it ever since, and love showing other people because it blows everyone's mind, as you've said many times, Carrley, and I'm sure, and all the people that have shared their stories, the same thing. So it's very cool, very cool. Brentwood Bay, paddled out to check out and swim in the bioluminescence, which was magical, but then got on top of my sup and headed to a very dark spot under the cover of some shrubs and trees looking down to the water. Very psychedelic experience. I felt like it was standing over the universe. Nice. Beautiful. That's really cool.
Yeah, it is truly magical. Mary is asking, she missed the first few minutes, but she's wondering what turned you on to study bioluminescence.
To graduate, I worked with it for a couple of years, guiding kayak tours, and then to graduate college, I needed to get an internship. And one of my professors said, I've got this internship studying bioluminescence from a kayak. And I said, yes, please. So that's what happened there. It fell into my lap, and I was pretty thankful for it. Graduation requirement.
Well, that's a pretty good way to fill your graduation requirement.
No, yes, yeah, absolutely.
Yeah, and sometimes people are always talking about like in the water, but I've had the experience of walking on a nice hard-packed beach out near
Mhm.
Tofino and rub your feet in the sand at night and it'll sparkle.
Mm. Yeah, it's so cool.
Mhm. Very cool. I've got one more question. What kind of techniques you showed the screen with the visual of your sample? Is there any special way you identify the bioluminescent cells, or it's just kind of like turn the lights off and figure it out, or is there anything special?
I wish it was turn the lights off and figure it out. It's kind of hard to make them glow in a lab setting. I never quite figured it out, but there's some good um, books out there, and I wish I had the one I used, but I used a plankton identification book and a lot of internet searching to distinguish this one round microorganism compared to this other round microorganism. So it took a long time, and they're known to be bioluminescent or not. So it just says this one is bioluminescent, this one's not. And again, it just needs more research. So just from books and identification guides.
Cool. Someone
One,
asks, sorry, John, I stepped on your toes there.
go ahead, Kylie.
How long do they live? Someone is asking.
Oh, good question. They can live from a few days to a few weeks. They're pretty fast, and they reproduce just by budding off of each other asexually. So it's pretty quick, their life cycles. And again, some of them are different than others, but I think it's worth mentioning this upcoming summer with the community boating center. The next person taking over the internship is going to study the comparison between bioluminescence and toxins in the sound. So he might have some more information for us this upcoming year. So.
Hey. Well, thank you so much, Carl. That was an amazing presentation. You were a
Thank
wonderful
you.
presenter, great storyteller.
Thank you. I'm still nervous, but that's okay.
Oh, well, it didn't show.
So I'm really pleased to introduce our speaker for the second half of our event this evening. So Salish Shah has more than 13 years experience working on the Salish Sea as both a captain and wildlife guide. In addition to managing the humpback whales of the Salish Sea project, she also works as an analyst for the Department of Fisheries and Oceans Canada. When not contributing to the Ocean Research Project, she spends time in her art studio creating elaborate illustrations about creatures who call the coast home. And again, there will be time for questions at the end of the presentation, and please enter them in the chat. We'll keep track of it. Thanks so much. So I guess, Tasli, you can go for it. One question, is that art behind you yours?
It is. Thank you for
Beautiful.
asking, Kylie. All right, well, here's the big pivotal moment, sharing the screen. All right. Oh. Okay, hope everybody can see that okay. Well, I guess good evening, everyone. This is really cool. I'm very thankful to the Transition Salt Spring Society for graciously inviting me to speak this evening, and to Joan and Deborah especially. I've watched almost every episode of this speaker series on YouTube, so I'm humbled and excited to be part of the conversation. And I only hope that I can be as smooth and interesting and dynamic as Carrley was during her presentation on bioluminescence, which was just fabulous. And admittedly, humpbacks are distinctly non-bioluminescing creatures, despite what you may have seen in Avatar: The Way of Water, but they are still really brilliant creatures. So I can't wait to talk to you guys about them this evening. So just a kind of a brief outline of what we'll cover this evening. I don't want to get into the really nitty-gritty of the humpback's basic behavior and ecology, as I mainly want to focus on the whales that we have in our region, the Salish Sea, but I thought it might be valuable to at least go over some basics before we move on. And the rest of the talk will follow suit, from commercial whaling on to the work that we do, And then a little bit of kind of lighthearted stuff at the very end, if anybody's still logged on at that point. But really, before we go anywhere, I just wanted to clarify what I mean by the Salish Sea. 'Cause at the time we started this project of cataloging humpbacks, the term Salish Sea wasn't even in regular usage. So I've created this map, this kind of hot Barbie pink. It shows what are generally to be the agreed upon marine boundaries of the Salish Sea. And generally speaking, we don't catalog humpback whales in the upper Strait of Georgia. And the reason for that is our colleagues at the Marine Education and Research Society, who I'm sure many of you are familiar with their work. They have sort of the cataloging of the whales in the upper Strait of Georgia. So again, just a couple of little review things. Certainly this will be review for many of you, but just a few basics before we jump right in. So humpback whales genus and species name is Megaptera novanglia, which is in reference to these amazing pectoral flippers that they have. They're actually the longest pectoral flippers of any whale, dolphin, or porpoise. They're approximately 5 meters long in some cases. And since these are the largest of the cetaceans that are regularly encountered in the area, we probably should talk numbers a little bit. So we're talking a maximum length of around 18 meters, which is about the same as a city bus, by the way, and a maximum weight of around 40 tons, which is close to 36,000 kilograms, which is about the weight of a fully loaded 18-wheeler. The females being a little larger than the males, generally speaking. Their lifespan is currently unknown, likely between 40 to 50 years, possibly older than that. There's no reliable visual age indicator after they're about a year old. It can be sort of difficult to class their age. There is a famous humpback whale on the East Coast in the Gulf of Maine known as Salt, and she's at least 48 years old, and she just had her 16th known calf in 2022. So just to give you a sense, she's 48 years old and still producing offspring. And their estimated daily food consumption can be, 3,000 pounds in some cases, which is about the weight of a Toyota Corolla. Speaking of their prey consumption, may be helpful to do this little compare and contrast with the other famous whales in this area. So humpbacks, of course, are collecting their prey from the water column by sort of skimming it or filtering it through their baleen plates, which you can see illustrated really nicely in this photo of humpback known as Columbia. Those kind of hair-like projections on the upper jaw is their baleen. So they're filtering plankton, which we heard a lot about in the previous talk, and small fish. And of course, contrasting with killer whales, which actually have teeth, which they use to grasp their prey. So these humpbacks are here in the Salish Sea to feed. That's why they're here. But what they're feeding on exactly is not really that well understood, but is currently being studied. So that's specifically their diet in this area. The trouble is they feed mainly at depth, not at the surface, where we can readily see or sample their prey. We can't assume to know what they're feeding on down at depth without some pretty creative research. Rhonda Reedy with the University of Victoria recently conducted her PhD study on analyzing fecal samples taken from humpback whales in both the Strait of Georgia and Juan de Fuca Strait. And these images that I have on screen are from her study, and they show bits of prey from the fecal material under the microscope, and her results showed krill was prevalent in 13 of the 14 samples that were collected in the study. So not really a surprise, but Pacific herring, hake, and eulachon were also prevalent, and sort of surprisingly, in one sample, sablefish was strongly detected, but it's not really known if this was sort of an opportunistic feeding event or if it's a species that the whales target often. And you may have noticed that there's a little feather in one of these fecal samples. Of course, this doesn't mean that the humpback whales eat birds. However, birds, particularly gulls and common mirrors, can be associated with feeding humpbacks, especially when the whales are targeting schooling fish at the surface. And this can be a little hazardous if you're a bird. So I just wanted to share this series of images of Hammer, who was busily feeding off of race rocks. And you'll notice here these red arrows pointing to these wings sticking out of his mouth. So Hammer would have come in, engulfed the school of fish that the birds were working on. And unfortunately, these gulls became entrapped in Hammer's mouth. You can see in the background, there's common mirrors that managed to escape. And before Hammer submerged, they did kind of a pause to expel all of these birds out of his mouth. It looks like there's at least 4 or 5 of them in there. And I've indicated that in the process of expelling the gulls, Hammer also spat out a few little fish. I don't know what species these would be. So you can see the birds just managing to escape here, and some possibly not. Very occasionally birds will be swallowed by the humpbacks, and then they are expelled as so-called bird bricks out the other end, but this is considered quite an unusual occurrence. Almost always the birds get out of the way in time, and luckily for the gulls and the common mirrors in the Salish Sea, since a lot of the feeding behavior occurs at depth, the birds don't usually come into situations like this very often. Okay, more basics, but still interesting. How can we tell males from females? Well, believe it or not, you can actually tell which one of these whales is a male and a female just by looking at these images. And I'll just ask if you can spot the difference. And I definitely don't blame you if you can't, because the differences are indeed subtle. You can see there that red arrow indicating a small lobe, which tells us that this is a female humpback. And that's pretty much it. All of their important reproductive organs are tucked inside of their bodies until they're needed. There's really no readily distinguishable physical characteristics between male and female humpback whales when viewing them from a vessel. We kind of have to get really lucky to acquire a high resolution image such as these two showing their genital area to confirm the sex. Or of course, if you document a whale with a calf in tow, you can pretty much assume that that's a female because the males don't associate with their offspring. And of course, DNA samples being the gold standard for determining sex. But the majority of the whales in our database are of unknown gender, but it's really not that surprising given the difficulty of knowing these things. So you can imagine my enormous excitement when analyzing this particular image of humpback whale known as gibbous, which allowed no doubt as to what sex he belongs to. This is a very rare image to capture, and this particular photo was taken during a very interesting interaction. It was gibbous and another whale named Scratchy, and they were interacting with a known female named Dreamer, and there was a lot of surface activity going on. All three animals were slapping their pectoral flippers on the water and breaching. It was a lot of posturing type behavior, and we very well might have been witnessing competitive type behavior between Gibbous and Scratchy for the attentions of Dreamer. And I believe having this photograph in particular kind of lends credibility to this notion, but we have a long way to go in understanding the humpback whale's social dynamics in the Salish Sea. Okay, last few basic things and then we'll move on. So humpback whales are a migratory species, of course. They kind of split their year sort of like 60-40 between a high latitude feeding ground, which would be a place like the Salish Sea, and then a low latitude breeding and calving ground. And I kind of put together just this little map, just giving you a general idea that once they're done their summer feeding season in the Salish Sea, the whales here will migrate to Mexico or Hawaii, and in a very few rare cases, actually all the way to Central America. They only stay with their mother for about 1 year, so they don't have long-term maternal relations like the killer whales do, for example. They're sexually mature at around age 5, though different humpback whale populations may have different calving rates. We actually last year recorded our youngest mother to date so far. That was Olympus, and she had her first calf at age 5. And they'll have a calf once every 2 to 3 years typically, although back-to-back years do happen occasionally. And the big one, I think, is that these animals show site fidelity. So they return to the same calving ground that they were born, and they usually return to the same feeding ground that their mother introduced them to. And they do this year after year. They become skilled and knowledgeable fisher folk, basically, with preferences as to where they feed and how they feed. And this means, of course, that we're not just seeing humpbacks passing through the Salish Sea going up to Alaska. These are known individuals. They come here year after year, and sometimes to very specific places. For example, there's certain whales that you're way more likely to see offshore Victoria than you are off Comox, and vice versa. Okay, this isn't a very good slide, I apologize, but I just really quickly wanted to introduce the concept of population stocks, or distinct population segments as they're known in the sort of management world. And our understanding of humpback whales is constantly evolving. And it's challenging to tease out the sort of hazy boundaries of these stocks, but just knowing that the whales show site fidelity and that their populations can kind of be broken down by the basis of movement patterns and genetics. And some stocks are more vulnerable than others, such as that Central American stock that I mentioned a moment ago. So we talk about a humpback comeback in this region, but why are they coming back in the first place? Like, where did they go anyway? I think this picture kind of tells the story pretty clearly. This is an image from Nanaimo taken in 1907, where there was a whaling station in Piper's Lagoon. And that particular whaling station took at least 97 individual whales from the Strait of Georgia in about a 3-month period. So just for perspective, in 2002, we recorded 160 individuals in the upper Strait of Georgia. So if those whalers were out there working today, they would have eliminated more than half of those whales. So it really gives you the sense of how rapid this decline was because of this harvest. Whaling data compiled by DFO indicates that at least 5,600 humpback whales were taken across the coast at the 5 main whaling stations. They were really exploited to sort of near total absence in this area, that for most of the 20th century, there weren't really any humpback whales here. And the only reminder that we had of their presence were names like Blubber Bay on Texada Island and Whaler Bay on Galliano, Whale Town, Whaling Station Bay, Balanis Island, like pretty on the nose stuff. So fortunately, in 1965, the International Whaling Commission got a little more serious, and they banned the commercial hunt of humpback whales in the North Pacific, which allowed this sort of new chapter to begin. And even just after a few decades of being untouched by whaling, the species began to show signs of rekindling their former glory. And reports of these 2 humpback whales roaming around Puget Sound in June and July of 1988 was kind of a hint at what was to come. So the sightings of these 2 whales in Puget Sound that summer was unusual enough that it warranted published research by John Calambakidis, who's a very well-known and respected humpback researcher in Washington. Nowadays, a few rogue humpbacks in Puget Sound is just kind of par for the course, but this particular event in 1988 really indicates just how absent they were prior to this. And I kind of like to think of these 2 whales as pioneers, starting the process of reclaiming their former range. To my knowledge, these 2 have not been seen since 1988, but others would follow suit. It was kind of the mid to late 1990s that humpback whales started to become a more regular occurrence in the Salish Sea. And as the legend goes, in 1997, this whale, Big Mama, showed up. At least it was our first time that she appears in our records. And my collaborator on this project, Mark Mallison, first saw her off of Victoria. He took a photo of her tail fluke, which has allowed us to identify her in subsequent years. And we've seen her here almost every year since. And her return each spring is a celebratory occasion by the local whale watching community. She had a calf who we call Moresby in 2022. So she might very well show up with a calf this spring, like in the next couple days or weeks. So we're all like eagerly awaiting her return. But she's very iconic in that she was one of the first whales to be resighted here on an annual basis, and bringing 6 confirmed calves with her in her time that we've known her. So it was really Big Mama and about half a dozen or so other whales that were documented by Mark in the late 90s that kind of laid the foundation for this whole project. And I know this is a ghastly slide with a terrible, boring graph, and I promise it will be the only slide like this. I just wanted to flash it up quickly to show that our project focuses on encounters where the ID of the individual humpback is known. So we don't collect just sightings of a humpback whale. We only keep them if we're able to identify the individual. So this figure shows how many unique individuals we have documented each year between 2003 and 2022. And you'll notice, whoa, what the heck happened here? Big jump between 2015 and 2016. And this does represent the year where more consistent effort on our part began, specifically in the kind of western reaches of Juan de Fuca Strait, where it can be relatively easy to find large aggregations of humpback whales. But this spike also follows a general trend of increased humpback reports from other groups who document humpback whale sightings, such as the Oceanwise Conservation Association and their BC Cetacean Sightings Network, which you can see on this graph. Again, this is from Oceanwise and their sightings network, and number of humpback whale sightings in the Salish Sea. And you'll note a very similar trend around 2015, 2016. The number of sightings really kind of went through the roof in a big way. So the North Pacific humpback whale population's recovering. These whales are starting to show up in our area. How do we catalog them? Or maybe a better question is why? Why would we catalog humpbacks? Like, why go through all this trouble? This might seem like a stupid question to most of you listening, but a couple of reasons why. Photo identification is often can be the foundation of projects focusing on cetaceans. And of course, the obvious parallel would be to imagine our understanding and study of killer whales in this area if we didn't catalog them or didn't photo ID them. It'd be very, very different. We can start to understand fluctuations in their population and in their distribution. We can look at their survival rates and their calving rates, and it helps increase empathy for these creatures, knowing that they're individuals and not just kind of automatons. So I would argue it increases care. And again, comparing with the southern resident killer whales, who are all kind of known individually. So with all of those things in mind, we developed a registry that records each individual's life history, their sightings history, points of identification, their kind of biopsy and tagging history, and evidence of entanglement and ship strike and so on. And we have just over 1,200 confirmed individuals in the registry. So this is sort of an example of a whale's profile. And you might notice that in addition to the tail fluke, we also catalog these whales using their dorsal fins and their tail stalks and their pectoral flippers. And the reason we go kind of all out on this is having as many points of identification as possible allows us to identify as many as possible, because frankly, you don't always get the greatest picture of the whale's tail, and they don't always fluke up. So meaning they don't always lift their tail when they go on a dive. Giving each whale an ID number is paramount in keeping things organized and logical for collaborative purposes. And our project categorizes and catalogs using methods developed by Fisheries and Oceans Canada. And they devised this kind of handy dandy XYZ system. So based on the amount of white pigment on the fluke. So it's probably fairly straightforward looking at the slide here that the X's are mostly black on their tails, Y's are sort of intermediate, and Z are quite white. And just as an interesting aside, the whales with mostly dark pigmented flukes are generally more common in the northern hemisphere than in the southern hemisphere, where it's you're more likely to see a really white tail fluke. And so in our database, just over half, about 52% of the whales are these X tail flukes with lots of dark pigment, and only about 12% are considered those white tails. So using Big Mama as an example, you can see that she's an intermediate fluke, so she's a Y. and she was the 324th humpback with an intermediate tail to be identified on the entire BC coast. So her ID number, BCY0324, which is great, helpful, but a little robotic sounding. So of course, we give these whales nicknames. Other than simply being easier to remember, there does seem to be a power in giving these whales names that we humans can better relate to. And BC has a long history of giving whales nicknames, and I'd be curious to know if anyone has kind of a deeper insight into the psychology of this. The nicknames are acquired from various sources. Usually the names reflect something memorable about their tail fluke. And admittedly, this can be a little bit like looking at a Rorschach test, you know, some imagination is required. For example, this is Yogi, and the name comes from the Marine Education and Research Society for the bear esque face shape on the fluke, though controversially, I happen to see the bear's face on the right side of the fluke, but evidently he was actually named for the bear face-like marking on the left side of his fluke. So I'll leave it up to you guys to figure out where it is. And just a few more examples of these nicknames and their origins. This is Neo Wise. So he got that nickname because of this beautiful comet-like streak on his fluke, and he happened to be born the same year that comet Neowise was in the sky above the Salish Sea. And this is Shepherd, nicknamed for that little shepherd's crook marking on the tail fluke. And I didn't know that Tutankhamun's burial vessel, he was holding a shepherd's crook, so I couldn't help but use this image as an example. And this is kind of cute. This is alto for the little alto saxophone shape on her fluke. And given that we're starting to document females with multiple calves over the years, we've started nicknaming these maternal lineages based around a theme. So this would be Ghost, and her offspring are all named for different words for spirit or ghost or something similar. And sometimes they'll get nicknames based on the shape of their dorsal. So this is Baker, and you can see that sort of amazingly that the dorsal shape resembles the outline of Mount Baker and neighboring peaks. So by collecting this data with confirmed ID information, we can observe things like calves returning after their first year. So remember, they're only with their mother for about a year. And then by the time they're 2 years old, they're on their own. So it's always exciting to actually see them, like, again, without their mother, and watch them grow, and indeed if they survived. So here's just a couple examples of 2021 calves that we've seen in each of the subsequent years, and we'll be eagerly awaiting their return this year. We actually had a big year for calves in 2022. We had 38 confirmed offspring, which was really exciting. And so far we've confirmed about just under 65% of those subsequently. And I mentioned this a little earlier, but last year we had 2 fairly young mothers. So Seabird had her first calf at 6 years old, and Olympus had her first calf at just 5 years old. And I just wanted to zoom in on Olympus and her calf, who we've been calling Achilles. So this is Achilles, sort of a lovely, precocious individual. I particularly like the image on the lower left where you can see mom is diving down, her fluke's up nicely, and then there's Achilles just off to her right, kind of doing a bit of a weird side angle dive. It sort of looks like he's doing a killer whale impression. His fluke sort of looks like a big killer whale fin there. Now, notice the very interesting markings that Achilles has on the fluke. And anybody care to guess what these might be? Well, we call them rake marks, and if you guessed that they're from killer whale teeth, then you are correct. Achilles survived a killer whale attack, which is amazing to think of. This would have happened on the northbound migration up from, I think these guys, this particular lineage goes to Mexico. So they're migrating up the west coast of North America. She's a brand new young mother, first time doing this, and they would have been ambushed by killer whales at some point. And mom, Olympus, managed to ward them off, and her first calf survived. And here he is, looking pretty cool. And before I get caught by any of the keeners out there, I know that this is a photo of K43, so not a whale that's going to attack a killer whale, but it's my only picture showing killer whale teeth, so I had to use it. These predatory interactions between killer whales and humpbacks are rare to observe. But in 2008, research by Cascadia Research Collective noted that about 15% of the whales they analyzed across the North Pacific had these rake marks. And without formal analysis, I can tell you that about 10% of the whales in our catalog have these rake marks. So that really represents like a real threat to humpback calves. But at this point, the big killer whales in the Salish Sea don't seem to bother with the humpback calves, likely due to the other far more easily targeted prey, such as harbor seals. I mean, a 40-ton humpback whale mother is not something to be trifled with. Though amazingly, we do see humpback whales and killer whales interacting here, though not in the way that you might expect. This is exclamation, the humpback exclamation circling around T49C, who happened to be feeding at the time. So in this instance and others like it, the humpbacks often approach bigs that are feeding and exhibit trumpeting behavior, which is like a loud exhalation, or they'll slap their flukes or their pecks on the surface. And the killer whales often seem a little ambivalent on these big whales coming in and raining on their parade, and they're generally the killer whales don't fight back in any kind of manner. Just a few more images here of humpbacks and killer whales interacting. But this picture is so cool, I think. This is humpback whale Nike, right in between T49A3 and T19C, and it kind of looks like they're traveling together, but what was actually happening here was these bigs had actually just chased down and killed a minke whale, and they were feeding on it, and Nike showed up and seemed quite aroused by the whole thing. And we also keep track of associations between individuals. So often these whales are encountered solo or in small groups. They're not known to show any kind of predictable pattern when it comes to associating with their mothers or their siblings after their first year of life. Like once a calf is weaned, that's generally considered to be the end of the association with the mother. But we keep track, and there are instances where offspring are with their mothers after their first year. So this is just one example in this image, Europa, the mother in the middle, with Callisto, who we're going to meet again in a moment. Callisto on the left, her calf from 2021, and then Eeyore was actually her calf from 2019, and the three of them were associating together here. And it's impossible to say if Eeyore recognized that this was their mother, and if that caused any kind of if that had any consequence on the interaction at all, totally unknown. But this is split fluke and divot. These are two known females. They're at both adults, and they were associating consistently for several weeks on end last summer. And they also showed a persistent association through 2015 as well. And these two are part of kind of the two big dynasties in the area, Big Mama's lineage and Heather's lineage. So they might relate to each other just based on the simple fact that they've been around in these waters for longer than some other whales. So there's maybe a familiarity or a bond there between them. But these associations between individuals really excite me, as we can now say with confidence who the whales are, and if there's more than one together, are there patterns to these associations to be teased out of the data, or is it just totally random who they associate with? I hope we'll know that in the near future. Oh, so I did mention that we cataloged their pectoral flippers, which are all unique as well. And that came in really handy, by the way, when last summer this video was getting traction on TikTok. And for all the wrong reasons, of course, this was seen as like a cute, fun video, but this was taken from the deck of a BC ferry, and I'm just going to play it, and I think you'll see that it's not actually a very fun video at all. Okay, so if you caught that, this is a humpback breaching extremely close to a BC ferry, and of course the identity of this whale was really important to figure out because we wanted to know if it survived this collision. And the only way that I was able to identify this whale from the video was through the pectoral flipper matching. So that's why we keep such detailed records on these guys. And I'm happy to say that the whale in the video, Callisto, was seen in subsequent weeks after that video was taken, so it did survive that near collision with the ferry. I'm aware of the time. I'm going on and on about humpbacks, so I think I might sort of skip through some of these slides that I have prepared. I think I'm gonna just skip to this one here. So I just wanted to show this overall kind of sightings hotspot map. And what this humpback comeback means for their conservation in the region. One of the looming threats is the increase in large commercial shipping traffic through the region. And this, maps like these can kind of give us an idea of where humpback encounters are more likely to take place, and unfortunately how these can overlap with the major shipping routes that go through Juan de Fuca Strait and the BC ferry routes. And recent published research from Christy McMillan at Department of Fisheries and Oceans investigating the kind of migratory destinations of humpback whales in BC has actually linked 13 individuals to the endangered Central American population, 7 of which show site fidelity to the Salish Sea. So making it even more imperative that we protect these whales because we have individuals from endangered populations. So speaking of migration, almost all of the whales on the BC coast go to either Mexico or Hawaii. Only about 0.4% have been documented in both breeding grounds in their life, and even more rare is for them to visit both breeding grounds in a single year. And one of those very rare individuals is our very own BCX 0767, aka Flint. One of my favorites, which is forbidden to have favorite whales, but anyway, he's kind of my favorite. So he was matched between Mexico and Hawaii in a single year, and he basically traveled between these two locations in 49 days or less. So really highlighting that the delineation between the breeding grounds may be less obvious than we previously thought. Okay, and just to finish off, the future, this is a fun talk to give because it's all very exciting. The whales were gone and now they're back, and they're just so lovely, and we get to learn about them and get to know them better and watch their family dynasties grow. But you may have heard in the news lately that the North Pacific humpback whale population declined by approximately 20% in recent years, which was associated with the marine heat wave. You might have heard it referred to as the blob. So that was kind of 2014 to 2016. And this knowledge of this population drop came as a result of a huge international collaboration helmed by Ted Cheeseman and his platform Happy Whale, which some of you might be familiar with, which is this machine learning tool using AI to identify humpback flukes on a mass scale. Generally, this decline seemed to impact the whales that migrate between Hawaii and Alaska, or northern BC, more than the whales that are kind of part of this like Mexican bound cohort. And as recently as 2022, the humpbacks in the North Pacific were assessed by COSEWIC, which, if I'm getting this right, the Committee on the Status of Endangered Wildlife in Canada, assessed the humpback whales in Pacific Canada to be of special concern. So they're not really out of the woods yet. And the primary concern that the report cited was this increasing frequency and intensity of marine heat waves. Okay, again, I know I'm just about out of time, so I think I'm just gonna maybe just try and get through this quickly, but just to wind down the presentation, I wanted to speak really briefly about the cultural impact of humpback whales. Early humpback whale songs recordings had a lasting impact, not just on the scientific study of humpback whales, but also in the public imagination. Humpback whales produce really complex calls that they use on their breeding grounds. And when these recordings were first made in the late 1960s, they were actually turned into an album for commercial sale, which did very well, by the way. And to this day, it's still Billboard's best-selling nature record of all time. So this is very captivating to the public. And I think the humpback whale song has become really ingrained in our cultural idea of whales generally. And it's humpbacks that produce these songs. No other species makes songs like the humpbacks. So these are just a few early examples of what humpback whale song inspired. So Carl Sagan and his Golden album, which was affixed to the side of the Voyager 1 space probe, which was meant to be this kind of acoustic record of life on Earth for any extraterrestrial that were to encounter it. The record had human greetings in various languages, different sounds from Earth like waterfalls and the sound of a human kiss, and there was Beethoven and Chuck Berry and the humpback whale songs. So they are literally interplanetary delegates representing our entire planet. There was an entire Star Trek film based on aliens trying to communicate with humpback whales. Highly recommend that one. Humpback whale song are used in movies and for sound effects to make kind of an ethereal quality, such as this Ridley Scott fantasy epic Legend, where they use humpback songs for unicorn calls. Also a great film. But most importantly and notably, the impact the early humpback song recordings had on the blossoming environmental movement can really not be understated. Especially Greenpeace's Save the Whales campaign, and some scholars even link the release of the songs of the Humpback Whale album with the beginning of the Save the Whales campaign period. And evidently Greenpeace actually blasted the humpback songs over the the PA system when they were chasing down Russian whaling ships and an attempt to get the Russian whalers to stop whaling, I suppose, an appeal to emotion. And the Animal Welfare Institute played the songs for the American Congress during closing arguments in the US Senate during the creation of the Marine Mammal Protection Act. So humpbacks are a big deal. And when I was preparing this presentation, Deborah suggested that I might put in some of my artwork into the presentation, and I thought I would just briefly show you guys some of the ways that the humpbacks have inspired me in my own personal culture and visual practice. So again, I don't want to keep you guys longer than I have to. If you have any specific questions about the artwork, feel free to email me afterwards or leave questions in the chat. But I don't want to bore you too much with with all of these, especially since it's getting a little bit late. I will just briefly discuss this one, though. This was actually a mural that was done for Seabreeze Eco Tours in Steveston, and this is a almost life-size Big Mama with her calf, Moresby, and their entourage of sea lion and marine life friends. There it is, kind of in real life. And in closing, I just wanted to end with this with something that I find really kind of amazing and poetic. So this is an old artwork of the Orion, and the Orion was a Norwegian-built steam powered ship, purpose designed for chasing and catching whales, and it operated on the BC coast hunting whales for 10 years, including in the Strait of Georgia. It was ultimately sold for scrap in 1940 and out of commission for whaling in 1916, but that was the Orion then, and this is the Orion now. He's a male humpback whale who we've been tracking for 14 years, and I just have to remind myself how amazing it is that we can expect to see Orion each summer when not that long ago we wouldn't be seeing humpbacks, period. So, and last but not least, I'm so appreciative for all of your attention this evening that I would love it if you could help me name this whale. So there's a couple of ways that you can get involved with this. You can scan the QR code on the screen or visit the URL I have on the screen as well, but probably easiest is in the chat. There'll be a link to a Google form where you can submit your suggestions, nickname suggestions for this beautiful whale, BCY 1088, who doesn't have a nickname yet. So with that, thank you. Sorry I had to rush at the end there. This happens when you start talking humpbacks, but thank you so much for your attention, everybody. I really appreciate it.
There, Kylie, or should I go ahead?
Yeah, no, go for it.
I was gonna say thank you so much, Cassie. That was really, really wonderful, and yeah, everybody is commenting how how passionate you are and how much they enjoyed that. So we do have a few questions. We have time for a few. Does the US keep the same name, or does the US have a different naming or identification convention?
Yeah, so sort of locally, the organization that catalogs the humpbacks in Washington and sort of the American portions of the Salish Sea is Cascadia Research Collective, and they use a slightly different ID code system, but we have sort of a master list of all of the IDs so we can cross-reference. But yeah, it would be good if there was kind of one universal way, but there's not.
Yeah, sometimes those whales had multiple numbers, multiple nicknames, but as long as everybody knows what everyone is talking about, right?
Yeah.
Are you using boat and land-based sightings, or do you use drone sightings too? Continuing, you mentioned tagging. Is there evidence that any Salish Sea individuals end up migrating to Hawaii rather than Baja?
So the as for the where the sightings come from, the vast majority of the sightings data that we get are from the extremely passionate and dedicated professional whale watchers in the Salish Sea. So naturalists like Joan and myself, so a lot of that is boat based, and occasionally we do get sightings from shore as well. Nothing from drones. There's some pretty sticky rules about flying drones over whales in British Columbia, so it's actually for permitted folks only. And I hope that answers the question. And as for the tagging of the whales and learning where they migrate to, in a lot of cases, the tags don't last that long, that you can't track them for their entire migration. So a lot of, that's generally speaking, many of the matches between feeding and breeding grounds come from photo identification. And in the past, that was a fairly arduous task, because you'd have to literally cross-reference thousands of images of thousands of whales. But now with AI and this platform Happy Whale that I referenced earlier, it can make matches in an instant. It's incredible. And so that being said, the whales in the Salish Sea are kind of split. Like, some of them, maybe, I don't know if it's not precisely half, but some go to Hawaii and some go to Mexico. We kind of represent an interesting transition on the coast. If you go much further north than here, then the whales are much more likely to migrate to Hawaii. And if you go a little south of here, they're much more likely to go to Mexico, like Baja. But we have a little column A, a little column B.
So another question about how many humpback individuals are using the Salish Sea?
So in our data set, we have just over 1,200 individuals that we've cataloged here between around 1997 up to 2023. And roughly half of those in a given year will be seen.
You want to catch the next question there, Kylie?
Oh, this is an easy one. Where can people buy your art?
Oh, that's so nice. My website, which is just my first and last name. So yeah, tasleyshaw.com. That's very nice.
And it's only the male humpbacks that sing, correct? Yeah.
Correct.
I didn't know that. That's new to me. We have humpback whales over here too, but ours go to the Caribbean instead of Mexico.
Yeah, that's right. I've always wanted to visit the East Coast, actually. We have lots of nice marine mammals there.
Come on over in July. What is the global population? Another question.
That I do not know. I'm sorry, I don't have that information. Maybe as consolation, in recently published work, that article I referenced documenting the 20% decline, the North Pacific population is estimated to be about 26,000 individuals currently.
Nice. One more question here too is, where do the whales of Quebec or Cape Cod journey to?
Quebec and Cape Cod.
So that's like in the States.
Right. I wouldn't know. Although Cape Cod, that I'm assuming that's probably very similar sort of population stock as the whales that you would see on the east coast of Canada, so migrating down to the Caribbean.
Yes, that would be my guess too.
Okay, I was going to backtrack just to one more, and I don't know if you're going to be able to answer this for these people, but how or where can we find out which humpbacks migrate where?
So again, that'd probably be a good Happy Whales, a place that you can go check that out. They have individual profiles for the animals, and so if there's one that you see in our catalog that you're curious about, you can type their ID number or their nickname into Happy Whale, and if they've been matched to a breeding ground, they should pop up.
And is that one for just Pacific humpbacks, or is that global?
Global.
Nice. I think I might put that in my canoe newsletter next. It's a cool one that people might not know about.
Well, I think we probably should be getting to a wrap up time now, but I have to say people did stay on, Carrley, so they weren't bored and they did want to hear what you had to say. So don't worry about the few minutes extra.
Okay.
Yeah, just a final thank you to Carrley and Carrley. You both had amazing, wonderful presentations, very enjoyable. And thank you to Kylie from Canoe for being with us tonight, and to Transition Salt Spring for being our host.
And thanks everyone this is the last week of the Wonders of the Salish Sea 2024. Thank you so much for attending, for your participation. Thanks to all our amazing speakers. And keep in touch with us. You can follow us on Facebook or Instagram. You can visit the website, and you can sign up for our newsletter. So thanks again, everyone.